Hi, Annie.
Hello.
Aloha, everybody. We're just getting started. We're gonna wait a couple minutes just to let more people show up. Thanks for coming. Good showing already. Awesome. Maybe give it one more minute here. Okay, let's jump into it. Aloha, everybody. Thanks for coming to another Hawaii Aquaculture Collaborative Brown Bag. Today we have Jenica Lowell Hawkins. Um, She's been doing aquaculture for over 20 years. She's the research director at Oceanera, and Oceanera also just spun off, I don't know if that's the right word, a new company called Konalimu Company, which she's going to talk a little bit about as well. So we're super lucky to have her. Awesome person. Take it away, Jenica.
Thanks, Kai. Can you hear me okay? Great. Well, you stole my thunder a little bit, telling everybody about Conalino Company. That was going to be like my big ending slide. It's okay. It's not really that much of a secret. So yeah, thanks. It's so nice to see a lot of familiar names and faces. But my name is Jenica Lowell Hawkins, research director at Ocean Era, and I am a recovering fish farmer. I've spent the better half of, or more than half of the past 20 years working in tropical marine finfish aquaculture, mostly here in Hawaii. Uh, and was so happy to come back here and join Ocean Era in 2021 because of the opportunity to learn more about limu and to get to work with it. And so I'm going to be presenting some of our work today uh, with this mouthful of a title, Effective Nutrient Source on Growth, Biochemical Composition, Bacterial Community, and several species of macroalgae. This work primarily was funded by the Pacific States Marine Fisheries Commission, as well as several other sources that I'll explain a little bit later. Um, and I hope it can kind of give y'all a sense of what we've been doing here recently. And if you don't already love limu, which I'm sure many of you do, it might make you love it even more. So Ocean Era, our whole mission is to kind of do good for the ocean. We want to soften humanity's footprints on the seas, but what does that mean? Let's stop extracting so much from the ocean without giving something back. But ultimately, we know that we have to do stuff that makes economic sense. So we're not doing research for the sake of doing research. We're doing research with the idea that we can commercialize products or practices that would eventually lead to, economic good, social good, environmental good. So as I mentioned, we work with marine finfish, Cereola, the Kampachi. We work with Nenui as well, and also some uku. These are species in various, stages of commercial readiness. Offshore aquaculture permitting is something I spend a lot of my time with as well. But what we're here to talk about today is limu. So our work with LIMU began in like 2017 or 18 with the U.S. Department of Energy's ARPA-E Mariner program with the long-term goal of raising seaweed offshore for biofuel, like fuel stock. That's a long time in the future. So in the meanwhile, we need to learn how to grow the seaweed and how to grow it right and what we can do with it to monetize it. So we're looking at it for human food, animal feed, fertilizer, fuel, and starting to dig deeper into the microbes, the enzymes, and the biostimulant capacities of this stuff. These are the three species that we're working with primarily, and that I'll be discussing today, Caulerpa lentilifera, they're on the upper left, Ulva lactuca on the right, and Halimenia hawaiiana on the lower left. All of these were collected here locally in Kona. Some of them as recently as a year ago. Other stock we've had for about 4 years. So we collected the stock, we brought it to our facility here in Kona, made sure it was clean, and we've kept it going vegetatively ever since. We have done some sporulation work with ulva, but that's the only one we've gotten to really start to dig into the sexual production of it. Everything else we've just done with vegetative propagation techniques. So the first trial I'm going to talk to you about is looking at finfish effluent's effects on seaweed growth. But the work here, it's not one project or one trial that I'm going to be talking about. It's projects and trials and experiments that happened over the course of like 2.5 years. For might be one species, it might be 3 species, but what we have here from the Pacific States Marine Fisheries Commission was the funding to do all this additional chemical analysis or proximate analysis. So certainly, if there are any questions, if you need any clarification, feel free to ask. Otherwise, I'll just keep on plugging through, and hopefully, you'll get a good explanation of what's happening. So this is work that we started in very late 2021. This is our facility here in Kona on the Makai side of it. The tanks in the foreground are where we had LIMU. The tanks just behind those black HDPE tanks, are where we held the fish. Here is a more detailed kind of schematic of that. So we had the fish in the 3 tanks in the back, the 3 yellow tanks. And then we had blocks of these 200-liter tanks set up. So the blocks of tanks on the left-hand side received 100% of their water from the fish effluent. So it went from the fish tanks into the LIMU tanks. The block in the middle had a mixture of surface seawater, which is of course very low in nutrients, and the finfish effluent. So it was a 50-50 blend. And the one on the right simply received water that came straight from the ocean, went through our filtration system, and then went into the LIMU tank. So very little nutrients at all. The species were randomized within each one of those blocks. So these were 200-liter tanks. They received about 3 liters per minute of seawater. That equates to about 22 turnovers per day. The fish that we used to provide the effluent water were the Cereal rivoliana, the Hawaiian kampachi that we got from blue ocean mariculture. We held them at a density of 5 kilos per meter cubed. And importantly, they receive a really high protein diet. So their diet is 45 to 48% protein, at least it was during this time period. And with a lot of protein going into the fish, that means a lot of nitrogen is going to come out of the fish. The surface seawater, like I said, very low nutrients, 4.5 micrograms per liter of ammonia. And ammonia is the compound of interest during this trial. But the finfish effluent water, the straight effluent water, had about an average of 100 micrograms per liter ammonia, so a good step up. So these ran for about 10 weeks. And here you can see the average mass of Caulerpa in the various treatments. So the blue line is 100% effluent water, orange line, the 50-50 blend, gray line, just surface seawater. And clearly there's a big difference. If it got 100% effluent water, makes sense because there's a lot more nutrients there. So it increased quite significantly. Interestingly, with Halimenia, we saw the same trend, but a little bit different. It did better in the blend of surface seawater and effluent water than it did in the straight effluent water. And of course, then in the surface seawater, it didn't do much at all. That trend continued through ulva, where again, the 50% effluent water had a higher growth rate, although I want to take this one with a bit of a grain of salt. The ulva was sporulating, and some of the biomass was hard to collect because it started to grow on the tank wall. So this was one of our first trials doing this, and we kind of learned a little bit, got a little smarter as we went on, so we wouldn't have the problem with like losing some of the biomass. But certainly the trends ultimately are what you would expect: more nitrogen, greater growth. Also, there was major differences visually. It became really clear the Halamenia became very bleached out without enough nutrients. The ulva would just get really spindly, and again, kind of behaved exactly as you would expect. Here's just something to explain it in tabular form. If our incoming water, which is that second column from the left, incoming total ammonia nitrogen when it was 100% effluent, 50% effluent, then 0% effluent, so the surface seawater. So that's what it's coming in at. Then moving over to the right, the Caulerpa, with 450 grams of Caulerpa in a 200-liter tank, it was able to remove 58% of the total ammonia nitrogen from the 100% effluent water. And that's how it's kind of explained here. So you see the Caulerpa was able to remove over 50% of the ammonia from both of the treatments that had effluent water in it. Similarly with Halamenia, 42% of the nitrogen was removed. 54% from the 100% effluent was removed. And then Ulva was, as we all know, Ulva was like great at sucking up nitrogen. It took up almost 70% of the nitrogen that was in the finfish effluent water. And this it corresponds to work by a lot of other researchers around the world. And then here's a little bit more of the data just to show some of the results we got from tissue analysis. So carbon levels, percentage of tissue, sorry, percent of carbon in tissue with the various seaweeds and the various treatments. So Fe is the finfish effluent. Then the middle is always surface seawater and effluent, and then to the right, seawater. Looking at nitrogen, again, interestingly, but not necessarily surprisingly, the more nitrogen that was in the water source, the higher amount of nitrogen that was in the tissue, except for Caulerpa. Caulerpa kind of stayed the same throughout. And again, the results that we see here with carbon and nitrogen ratios correspond to other folks around the world, where you're going to see a lower ratio as your nutrient inputs change. Additionally, with this work, we did food safety testing. So we looked at fecal coliform in the water, and we found that fortunately, there was none of that. And we also did some elemental analysis. And if you guys want some details, get in touch with me later and I can share them with you. We looked at all these different elements to see if there would be different amounts of them based on the water source. And there certainly was with some of them. I don't think any of the levels ever came to a point of concern, but definitely there were a few cases where the effluent water either had a significantly higher or lower amount of the nutrient of interest, or excuse me, the metal of interest in the seaweed. So that's something that we always need to consider. And just as an aside, we continued doing food safety testing in 2024, and we might talk about that again when we talk about Kona Limu Company, as we need to start doing a water testing and food safety program to sell to some of our interested buyers. This is a totally separate experiment. This is one that we actually grew here, performed here in our lab, looking at the nutrient supplement F over 2 on Halamenia and how that would affect growth and also the uptake rate of some of the nutrients by the Halamenia. So what we had here, we had flasks of Halamenia in the lab, and these flasks were given either one treatment of F over 2 each week, 2 treatments of F over 2 each week, or 3 treatments of F over 2 each week. And as you can see, we have the weight of the Halamenia each week. And there was really no difference based on the number of times we applied the F over 2. This stuff, honestly, it's just so concentrated. The Halamenia was able to probably uptake what it needed with that first treatment, and the additional treatments weren't really necessary because there is no significant difference in final weight based on the amount of treatments of F over 2. Then with some extrapolation, we could and some water chemistry testing, we could figure out the uptake rate. And so that's listed down here in the table at the bottom. And ultimately what that told us was a gram of Halamenia can take up about 0.5 milligram of nitrate, nitrogen. I'm not going to try to say that again, in 1 hour. So again, this is just some extrapolation that we did, but that's a gram of Halamenia can uptake 0.5 milligram of nitrate over the course of 1 hour is kind of what we were able to ascertain from this. Now, another thing that we were looking at with this funding, with these projects, was how water sources affect the microbiome of the limu, because that is an incredibly fascinating field of study to get into. And especially those of us that are like fish biologists, and I have a background in animal health, this stuff kind of like blew me away. So we did this work in conjunction with Craig Nelson at UH Manoa. And we looked at, again, like you can see here, 115 samples, a bunch of water samples, a bunch of limu samples, and we found so many different things. And we don't need to like get into the details of this fun little heat map, but ultimately what it tells us is the microbes in the water are very different than the microbes in the limu. The limu does an incredible job of being able to control its own microbiome and its own bacterial community, despite whatever might be coming in with the water source. So again here, so whatever these guys are on the left, upper left, really lots of those, lots of that bacteria there in the water, but then as you move to the right, none of it there in the limu. And get into this in more detail. So here, looking at our different types of seaweed, the Clerpa on the top, going down to Ulva, Agardiella, Gracilaria, Guadalupia, and Halaminia, we can see the types of bacteria that each seaweed tends to hold on to. So the Clerpa, obviously, whatever the, and I don't remember what they are off the top of my head, whatever the bacterial families are that are that deep purple color and green, that's primarily what it's seen. And that doesn't matter if the water comes from deep sea water here in Elha, surface seawater here in Elha, if it's finfish effluent water, that's those trends that like one group of bacteria is consistent throughout with the Clerpa. The one that I've always found the most interesting is the Halaminia, and it's shown down there in the bottom. Is one thing we've always noticed is that Halaminia has very little trouble with fouling, unless it's fairly stagnant water and the Halaminia is very bushy. Virtually nothing else will grow on it, whereas with some of the other species, they can be taken over by epiphytes. And if you look here, you can see that there is like 4 types of bacteria that is commonly associated with the Halaminia, and that's so consistent throughout. And it was really fascinating, which again, it kind of corresponds to what other researchers in Southeast Asia and India have been finding, that there's some unique maybe antibiotic properties or other methods of bacterial control that the Halaminia is able to do. And that's got to me, and I think about the implications for health and therapeutics development. It's really fascinating. So here, looking at how do the water sources affect the type of bacteria that we're going to see. It doesn't really matter. The nutrients didn't seem to enrich the different microbes, and so here again you'll see the different types of bacteria are associated with different colors, and the sources of the water are there on the left. The trends are pretty standard, except for the deep sea water, which makes a lot of sense. Everything else here, the effluent source water and the finfish effluent, they kind of all stem from the same place. So again, this is work that Craig Nelson did. Some of the samples of water were not necessarily from our facility. Some of the samples of the seaweed were not necessarily from our facility, but the majority were. And anything from stuff that we're familiar with, like Vibrios, to certain plenty types of bacteria I've never even heard of before were examined in this. And again, if anybody's interested, I'd be really happy to share more of the details of the results if you want to get in touch with me. This was another bit of work also by Dr. Nelson along the lines of food safety testing. So we were looking to see if microbes that are commonly associated with the gut would be more prevalent on limu that was raised in finfish effluent. So we were looking at Gracilaria was the species we were looking at this time. The gut microbes that he examined are listed there on the left, Bacteroides, Vibrio, C. ultramonas, et cetera. And interestingly, by looking at the charts on the right, the blue line is the bacteria. It says filter, but basically that's the bacteria that was pulled from the water column. And the red line is the bacteria that would have been found on the limu. So as those lines move to the right, those are the percentage of finfish effluent that is in each water sample. So on the left-hand side, the start of those lines, that's 45% finfish effluent in that water column. And then it's 100% finfish effluent to the right side of that. And so again, not surprisingly necessarily, as the amount of effluent water went up, you'd see more of those gut-associated bacteria. But looking at the limu, that red line, no matter what was in the water column, it did not affect what was on the limu itself. So bottom, everybody knows Vibrio knows it can be problematic, and that's shown on that last one on the bottom. As the amount of finfish effluent went up, there was more Vibrio in the water, but it did not affect the amount of Vibrio that was associated with the lemur. And again, this stuff to me, as a animal health background, it's incredible the way this can control it. It's really fascinating, and it just shows the potential that this has. And one of the reasons we're so happy to be growing it and offering it as a food. I mean, people that are from Hawaii or from Southeast Asia, they know how good seaweed is. I didn't grow up eating it, unfortunately, but I'm sure happy I do now because it really has so much to offer to us. So that's the kind of overarching view of the sciencey part of it. So thank you to my co-authors, Simona Uite and Craig Nelson and Neil Sims, of course. PSMFC for funding the work, as well as the other groups that funded us, the Department of Energy, our partners at UH Manoa, World Wildlife Fund also funded that first project that I was discussing. It's really been a long effort, and we've done so many things to learn more about the stuff and learn how to grow it better. We continue to kind of do that every day. And so, yeah, what it's ultimately led to is this. We are starting a new business called the Kona Limu Company. I'm so stoked and so excited to get to share this with you guys. We've been selling a little bit of seaweed into the market over the years, but only within the past year did we really make a push for it. And it's really caught on. People really, really like it. The Caulerpa is beautiful. My 8-year-old runs into the farm and like grabs handfuls of it out of the tank, which is always exciting to me. The Halimenia is my favorite because it's so versatile. And Ulva, that one, it's beautiful. I haven't figured out how to cook it yet, but maybe you guys might have some ideas. But this is what we've been getting up and going, and we're hoping to expand here in the coming months and get the market to continue to grow. And with that, I guess I'd like to say thank you for your attention, and let me know if you have any questions.
Thanks, Jenica. Sorry, I actually don't think I stole much of your thunder. That was awesome. Thank you. I just had a quick question before we open it up. A little bit of a Limu question. Um, could have, if you looked at the graphs longer, done it. But I was wondering if you had calculated the percent daily growth rate of the Limu you had
Um, you know,
total.
we do. I don't have them presented here, but yeah, we look at a daily SGR with pretty much everything that we do. That again, be happy to share. I think off the top of my head, the Caulerpa grown in the 100% finfish effluent, we had sometimes we had SGRs as high as 9
Wow.
for that one. Which for that that was like a record. I don't know if we've been able to hit it since, but that one Halimenia, I think our best was usually around 5, but of course it would vary. But again, yeah, happy to share the details if anybody's interested.
Oh, that's great. Okay. Yeah, let's open it up. Anybody have any questions, Jenica?
And I can put slides if you want. I can put slides or pictures or graphs back up.
I think Jim.
Uh, Excellent presentation, Jenica. I thought the microbiome stuff was really fascinating, and but it was kind of quick, passed me quickly. And I'm just wondering, are you talking about like the microbiome associated with each of those limu in the water column, or was it attached to, when you showed that each limu had their own microbiome typical?
So that would be what is on the limu. So
It's on the limu.
some of the work did test like here's the scoop of the water from the tank, and here's the limu grown in that tank, and then they would test the microbiome on that limu, and then also in the water column.
Yeah.
And so it was always, that's how we were able to kind of show that the limu, if you were to clean it up before you eat it, you really wouldn't be getting anything that was in the water column, unless
Yeah,
whatever microbiome. Yep.
I'm just wondering, is there a another product in your mix that's not whole limu, but it's some kind of microbial extract from the limu that has these beneficial microbiome factors? Like, it almost looked like you had an anti-vibrio
And
component somehow in the red limu. There was no Vibrio there.
right.
What's that about? And
Yeah,
how is that something that's exploitable? Do you
sure. I mean,
know?
I can tell you what Dr. Nelson explained is the limu, they produce exudates. And it seems to be that which is able to kind of help keep some of those bacteria, I imagine, keep those bacteria at bay. Either the
Right.
community of bacteria can, push away the stuff it doesn't need, or whatever is in some of those exudates is able to do that.
So that's like a gene product, a protein or something that the lemur is producing is kind of, at least Dr. Nelson's thinking, that's what's controlling that microbiome. It's actual natural products coming from the lemur that are
Yeah,
affecting.
somehow the limu is able to manage that bacterial community, or the bacterial community and the limu like work together to kind of manage what's there. Um,
That is
but
super
how
cool.
I don't
Yeah.
either.
Wow.
But yeah, it's pretty
Super
awesome.
cool stuff.
Yeah,
Thank
Um,
you.
Uncle Wally.
Wally, I see your hand, but I think Anthony had his hand up first, even though is that right, Anthony? Maybe
I
we'll
don't know, but I can go first, I guess.
go for it.
Um, Thanks for that presentation. This is so exciting. There are a couple of different things that I'd love to just go one-on-one with you, Jenica, on. Um, but for sake of today, I was curious about, I just did a quick calculation and you reported your nitrogen levels for various species of nitrogen at 100 micrograms per liter, which is zero point 0.1 parts per million. And in the aquaponic world, that's extremely low. Um, and in the Limu world, to my knowledge, it's still fairly low. My question is, are you guys going to continue to do work on the marine aquaponics field and really kind of nail down the nutrient delivery? Because it seemed that you're reporting both nitrates and ammonia together. So there's no filtration step, no biofiltration step. Is that correct?
So actually, what we have here is I reported ammonia in one of the trials and nitrate in another trial. We were actually those were two separate things, and we are looking at them separately. And it really isn't. Unfortunately, these weren't the first project that I talked about the most was not recirculating, so it was still all flow through, which is one of the reasons probably the ammonia was generally low. And also that was an average, because during the day after the fish would eat, it would spike quite a bit higher, but then drop. But yeah, honestly, what brought us to do this was this work we're doing with the Department of Energy, because we wanted to see kind of how low can you go with your nutrient sources. Because if we want to try to raise seaweed offshore, you're never going to have a ton of nutrients near the surface. And so, what is kind of that minimum amount that we could have that would still get a decent growth rate, which is kind of why we were okay with exploring these kind of lower levels.
Got it.
Um,
Yeah, that's a different target. I see where you're coming from. That's awesome.
did that answer your question? Was there another part to the question I
Yeah,
didn't?
yeah, no, the approach for the project is just
Hmm.
different than, yeah, what I would have
Yeah.
thought. So that makes sense. Thank you.
Thanks, Anthony. Wally.
Oh, thanks. Sorry, I want to go back to Jim Wyban's observation, and I was at kind of the same thoughts too. And I just not so much a question, but a comment. And this is my personal observation. We're working with the lemur species. The the hulu hulu vina, or the Guadalupia, and the what is that, Halaminia. Sorry, I'm thinking Hawaiian names. The Lepiohina
That's okay.
both have like a real slippery texture to it. In fact, that's one of the ways that we kind of like identify, especially the hulu hulu vina. You just feel with your hand, and you feel that slippery texture. That, oh, this is definitely hulu hulu vina, yeah. So I was thinking that maybe something on the surface that makes it. So adding on to Jim Wyban's observation, maybe like taking that surface microbes and seeing what's happening there. Thank you.
Thank you for saying that. And but honestly, I we've been trying to grow the hula hula vina, and we have not had great success because we've had problems with fouling. And that, thank you for sharing, because that's a great clue that we're not giving it what it needs. If traditionally you've seen something differently, because that's actually a struggle we've had with the little bit that we've had here on site.
Ame.
Uh,
Hi, Janica. Great presentation. and thank you for taking my question. So I had a few things, but the Halamenia, are you guys selling that abroad? Like, could I get samples here on Oahu? And do you, in tangent to that question, if I could be able to buy it here on Oahu or order it from you guys somehow in bulk and get it shipped over, have you guys run a nutrient profile on the contents of the Halamenia? Because I'm looking at its red pigmentation, so I'm wondering how much maybe anthocyanins or carotenoids does it possess?
Right. So we have surprisingly have not dug deep into like carotenoids or any of those. And we should and we need to and we would love to. We are if is Neil Sims in here because I'm pushing like we got to get to Oahu more and sell more of this stuff on Oahu. Certainly, if you know if it's a collaborative thing, of course we can send you some. Because we want to get the answers to some of these questions as well. Like how, if we are offering it more nitrogen, is it going to have more protein? Is it going to have, more of these compounds of interest? We don't know the details. We've done a little bit of work with proximate analysis, and I think once or twice we might have done some, fatty acid profiles or amino acid profiles, but not as much as I would like to. So certainly we want to dig into that more.
Oh, okay. Amino acid profiles could be maybe helping along the way to, you know, um, indicate what may be present from the conversion ratios, like the
Okay,
quantities that you're measuring.
gotcha.
Interesting, interesting.
Yeah, we'd
Second,
like to do more of that for sure.
yes, yes, I would love to be looped into that because I'm very intrigued with doing research with microgreens. We just did a 2-year study on the contents of microgreens and impacts like ultrasonic treatment at the seed stage for cracking of the shell. Um, it's very interesting. So I'm very intrigued by those kind of like radical cell fighting, cancer cell fighting kind of characteristics of these kinds of plants that we may have locally that's nowhere else in the world. And so it has an environment that may be, benefiting in some way in those productions. But sorry, tangent. Second question was or not really a question, but going back to your analysis that showed that somehow the algae, maybe through a mucilaginous coating or something, is controlling the Vibrio colonization on their surface. I don't know if I kind of was away from and I was listening at a distance. So I think that's what I heard.
Yeah,
I might be wrong on that.
nope, you got it.
Okay, what species was that on? Was that the Halamenia? Was that the uva?
That was actually Gracilaria.
Gracilaria. Okay, okay. Intriguing thing that may have paralleled with or interconnected with some of the work I did at UH when I was managing the aquaculture lab. We often would see Vibrio breakouts in both the Penaeus venemi and in the Macrobacterium rosenbergi. So the giant river prawns and the white-legged Pacific shrimp. And those kind of Vibrio outbreaks, would require really strong medicated treatments if we were going to treat them, but we didn't do that in the lab. We didn't have permit for that. But if you could isolate how that mucilaginous or whatever coating it's using, to control that Vibrio, I think that would be an intriguing kind of research that you guys could maybe look into because you can generate the seaweed already, the limu, you can produce it. So maybe there's some kind of pharmaceutical or nutraceutical kind of interest there.
Yeah, folks are like I said in other countries and as well here in the US, but really starting to get into it to try to understand what we can learn from these different limu and how they can help us figure this out, especially stuff that is a biofilm forming bacteria like Tenacibaculum and even Vibrio. How is something like the seaweed that grows its own biofilm basically or exudes its own biofilm, how it can kind of combat that. Yeah, so it's there's so much we don't know. It really is, so how can we use it and use it intelligently, right? To kind of do good things with it.
Well, yeah,
A lot
could
of
you
potential.
raise the shrimp together in the same environment with your limu and then combat it that way? Or if the shrimp ingested the limu, would that impart some of the protections? It's
Yeah, as an
that's
ingredient is something, because we do a lot of fish nutrition research. So that's definitely something we've been looking at.
Jim is very knowledgeable about the Penaeus venemi. You might want to tap him for that. Just saying. But thank you very much for taking my questions.
Thank you.
Jen, I have another question, real quick. The others at NOHA have used the deep well water to grow limu with basically no input.
Mhm.
And I know that would throw a temperature issue into your experimental design, but did you ever think about using
Oh
that as
yeah,
sort of a control?
we use it a lot. I mean, today I shared maybe, three three or four kind of, trials that we had going on, but we've done dozens and dozens over the past couple of years. Absolutely looking at deep sea water, the percentage of deep sea water, if we mix it, 20% deep sea water with the rest, 80% surface sea water, how does that do with the different seaweeds? And yeah, so we do that quite a bit. That's actually how we grow our Halamina. We don't use anything else but deep sea water. The Clerpa, we've done a little bit of everything. It seems to some of the fertilizer products more, but all of it's very adaptable. Like it will adapt to the nutrient source that it's offered, certainly after a couple of weeks. But yeah, definitely look at deep sea water. And Halamina doesn't mind it being so cold. The Clerpa doesn't like it so cold. So that's the kind of stuff we're actually figuring out, again, as part of this greater Department of Energy project we're working on.
Right on. I figured you guys had been playing with that.
Anthony, I see your stuff in the chat. Yeah, thank you. We definitely look at Dr. McDermott's information a lot, and we've done some of our own as well. Yeah, there's some good stuff out there. So she's produced so much. That's been a great basis for understanding.
Anybody have any other questions for Jen? We got her.
Oh, the question is the funding from DOE impacted and business as usual? Uh, Not business as usual. The stuff we had, we continue to have. They didn't take it away. Decisions on anybody here that's been putting in grant proposals who knows they're not making decisions on it right now, and that's definitely the not business as usual part. So fortunately, we're still able to keep going with the current project that we have.
I think Anthony has another question. Go for it.
Hey, regarding food safety, I really am stoked to see that Limu have some sort of basal defense against, the more pathogenic bacteria species that we're so concerned about. And there's not, if you look around in the literature, but also just talk to anyone in food safety, we're not really at the level in Hawaii as I think we want to be at for food safety with seafood products. And so it's really exciting to see this. Have you been able to hack away at getting a certification for food safety, specifically with Limu?
Right. So we don't have one as of yet. We're actually just this past week started working, talking with some produce distributors to understand what their needs are. I mean, ultimately, I'm not sure how much certification is going to cost, but also there's the certification, but also just the steps you have to go through to get there. And if you look at it like any other, it's raw agriculture commodity is what seaweed's considered, so it's like produce. There's some kind of I think overarching steps we know we have to take, like maybe quarterly water testing for E. coli, salmonella, and listeria, and then having those, that HACCP type of thing in place to make sure that people are washing their hands properly and stuff like that. So we're making a little bit of progress slowly. I got some information just like yesterday from a potential buyer that would help us towards certification, or at least checking the boxes that they need to check. So yeah, we're kind of making our way down that road, and hopefully we can come up with something. We've also been helped by North Shore. Is it North Shore Economic Development or North Shore Education? That
Yeah,
Hui people that is helping to kind of do food safety and group gap stuff. So yeah, we're kind of working with a variety of friends to kind of help us go down that road.
nice. Are you guys able to, as you move along, kind of like help the industry in that capacity as a kind of a example?
I don't see why we
Like
wouldn't. Of course
maybe,
we would. Oh my gosh.
yeah,
Yeah, if it's something like this is what's worked for us, and would we tell people? Absolutely.
Yeah, because
Because
there's
a
a
lot of steps and processes, and it's just hard to work those out. But once we work them out, it would be similar for most people or most processes or most most farms and products.
okay. I'd like Dar to help with that or rope in DOA, maybe do some training when we figure out what the steps are. There's some schools that are, producing seafood and other places that I'd like for them to consider things that you're going through. But anyway, thank you so much. That's a good answer.
Anybody have any other questions, Jen? Oh, on me again. Go for it.
Thank you, Fox. Kai, sorry. Sorry, I was following up Anthony's comments and questions. I am currently working with a sustainability food, what did you call it? I know she teaches sustainability food systems or sustainable food systems over at Farrington. Um, and I was thinking that if they could generate limu or utilize limu that's generated locally in some of their processes and course material, that might be of value to them. Would you mind if I share your contact information with her and kind of like when Kai puts up the video for the media, I'll share the link with her so she can take a look at what the material is that you have and what it covers and see if she'll be interested in that. Is that all
Sure,
right with you?
of course, of course.
Awesome, awesome. Thank you very much.
I can, also do a quick update on that too. I believe, Jen, you folks are part of Casey's ongoing study for food safety. Um, maybe
We've
not.
definitely, yeah, no, I mean, we've definitely been working with Casey and
Yeah.
sent her a variety of samples. I'm not, I can't remember which, or if she used it for multiple things or if it was part of the food safety or not.
So she got funding from Department of Ag to do both micronutrient analysis, some of the other maybe heavy metals, that kind of contaminant, and then also the microbiological. So we're coming up. She's over the past year has been sampling monthly, like you were
Oh,
talking about, to
fantastic.
establish like geometric mean for a variety of different sites for lemur culture. So that hopefully that'll be coming out soon.
Oh, that's great. Yeah, because I mean, I'm trying, if I understand FSMA, so much of it's just going to be like make sure your processors are in place and make sure your source water is clean. And then we go from there. And then, of course, that gets followed up with like, very regular testing.
Right. Maybe one more question for Mommy.
Sorry, following up on that, Kai, are you guys and Jenica, are you talking about Casey Ho, Dr. Casey Ho?
Yeah.
That study I mentioned, I was doing that with her grad student with Taryn. So I work with her too. So, oh, okay. So she's in contact with folks, and you're doing the heavy metal studies or site verification study with
I
them.
don't. So with Casey, I know, and I've, my colleague Gavin has been in touch with Casey more than I have recently, and we've sent her a bunch of samples. She hasn't been here on site to get any samples, but certainly she may have integrated our seaweed into any of the tissue testing. Quite possible.
Yeah, she did mention to me there that she was working on some lemur research, but because mine's is I was assisting in microgreens, it wasn't connected. So she didn't tell me specifically. Oh, even more connection. I'm very excited to follow up more with your work. All
We
right, thanks.
appreciate networks. We really do.
Small island, right? Small state. We all gotta help each other. Thank you
Absolutely.
again.
Thanks, Ami. Rock, you had a question?
Oh, not a question, just a quick comment. Because I work with Casey on the Limo project too. So yeah, we can do heavy metal analysis on campus. Very nice talk, Janica.
Thank you.
Yeah. Thanks, Jen. This was great.
Absolutely happy to and really excited to like share this with everybody. Thanks for the opportunity, Kai.
Of course. And as usual, yep, like Ami said, we're gonna get this. We recorded this video. We'll get it up on our site, on our actions page at Hawaii Aquaculture Collaborative. But thank you all. Really appreciate it. See you next time.
Thanks.
And
Aloha.
aloha.